Hello everyone, and welcome to Essen Biosciences monthly webinar series. This is our second webinar, so if you've joined us before, welcome back. And if this is your first time, we're glad to have you with us today. So now without further ado, I'm very excited to introduce to you today's speaker, Vince Groppi, Vice President of Drug Discovery and Chief Scientific Officer at Essen Bioscience. Vince is an experienced molecular pharmacologist, drug discovery and development project leader with 27 years of experience in making a real impact in the fields of psychiatric, neurologic, and cancer discovery. The breadth of his experience includes working closely with all aspects of drug discovery, from assay development, high-throughput screening, lead optimization, in vivo efficacy, pharmacokinetics, regulatory toxicology, and identifying translational biomarkers for clinical decision-making, as well as broad experience in understanding the commercial and medical needs of patients with neurological and psychiatric diseases in all world markets. As I mentioned, Vince is currently the Vice President of Drug Discovery and Chief Scientific Officer at Essen Bioscience, which has really broadened his experience by working with all major pharmaceutical companies and many biotech companies on drug discovery and technology projects. He is a member of 2 standing NIH study sections and a member of the NINDS Blueprint Therapy Network. Vince's passion in his professional life is to drive to find medicines to improve human and animal health. And we're very grateful for his time today. So, ladies and gentlemen, Vince Groppi.
Thanks for that introduction, Jason. Thank you very much. And hello to everyone who's tuning in. This is one of my first webinars, so I'll do my best to make it seem like I'm in the same room with you. And as Jason said, we're here to transmit information to you that we're excited about, and we're also very interested in hearing your comments and questions. So please use them, and I'll address them at the end. I'm gonna basically tell you 3 things today. I'm going to tell you a very short little bit about Essen Bioscience and about how we position ourselves and what we do that we think is top notch and honestly is good or better than anyone else in the world. And then I'm going to give you an example of how we work together as a group within Essen and with strategic partners to answer important questions. And I'll end by giving some ideas about some future projects. So for those of you who don't know us, Essen Bioscience is a privately owned company. And we're small. We have about 80 FTEs right now. And we think of ourselves as very nimbly run. And because we I think have worked hard and chosen wisely, we've had rapid growth. So it was over the last 5 or 6 years. It was co-founded by 2 friends and colleagues, Kirk Schroeder and Brad Nagel. I've known them since the early 90s, and together we've put together a senior leadership team that has collectively about 100 years of drug discovery experience. I'll tell more about that in a later slide. Working together, we've had better than 40% revenue growth year on year since 2004, and we've been profitable every year. We have base of operations from a laboratory point of view in the US and the UK, and we have direct sales and technical support in all of North America, Europe, and the Pacific Rim. When I think of Essen, and I try to summarize for people when they ask me about Essen, I basically say what we do is synonymous with excellence, and it starts with excellence in engineering. The history of our first-in-class and honestly best-in-class instruments starts with Flipper in the early 90s, a project that I was directly involved with, and when I was working at the Upjohn Company, and then goes through the invention of Ironworks and lastly IncuSight. So that's our engineering excellence. And what goes part and parcel with that is the requisite reagents and assays that you need to take an instrument, use it to answer questions, and generate both information and hopefully at the end of it knowledge. And in thinking about it with the end in mind, the assays that we build, we stand behind them, and they're fully validated, both technically as well as pharmacologically, because we are quantitative biologists, and our assays are designed to drive decision making at all levels. That then builds the foundation for doing drug discovery. Initially, the tools that we provided were to end users, and drug discoveries were done externally. And what I'll tell you about is how we've taken that foundation and built it internally as well. So our core business is instruments, assays, and reagents. The assay that we currently manufacture and support is IncuCyte. It is an imaging system that is designed to work within a standard CO2 incubator, and it generates data that we call live kinetic imaging, and it's perfect for developing phenotypic assays. A snapshot of the assays that are built for IncuCyte are here on the second part of the slide. It started out as a cell culture QC tool, but has grown rapidly from that, and in the field first of cancer research. So the assay, the instrument set up to kinetically monitor proliferation, apoptosis, cytotoxicity, migration and invasion, my personal favorite, angiogenesis, a very difficult assay, and reporter gene functions. And something to note for this presentation is we have an assay that does a good job tracking the growth and branch points of neurons using a non-label technique. Happy to talk about that at some point in the future if anyone's interested in it. But you know, when it comes to doing anything, it really is, it's all about the people. And I would say that we're lucky enough to have among the leaderships of the team some of the best in the industry. And I'm not just talking about myself, I'm talking about folks like a recent hire here at Essen is Owen McManus. He's the director of the Ion Channel Discovery Group, formerly at Merck, which had one of the the best ion channel groups in the industry. So Owen's here and now is the director of discovery. Dave Rock comes from a background of Park Davis and Pfizer, and like Owen, was central to all of the great work that came out of those organizations. Deltra Zeiss and his close friend Tim Dale were the leaders of the ion channel group at GSK. So when you wind us all together, you see that we have best practices from GSK, Park Davis, Pfizer, Merck, Pharmacy, and Upjohn, and all of that is what we bring to bear on the questions that we choose to ask internally and the projects that we take on with external partners. So from that point of view, there's four ways of working with us, 4 modes. The first is simple consulting. We're happy to do that. We also do customized reagents, assay development, and drug screening in a fee-for-service manner. And we also get involved intimately in R&D projects when we can be brought in as a partner in the projects, and we have a system in place, a partner network in place, where we can prosecute a full R&D project all the way from concept to phase 2, and I'll tell you about that in a minute. So this is just a sort of an example of a graphical example of what I just said in words. So from our discovery services side, which is our fee-for-service side, we have ion channel assays, we have the live content phenotypic assays, and since we invented the system and have used them ourselves, we bring strong technical expertise to the table. We build that into a drug discovery network that includes partners that I'll tell you about at the end, and is based on essentially deep knowledge of the areas that we choose to compete in, and a strong partner network with equally deep areas for their own expertise. And what we pride ourselves on is developing customized solutions for efficiently and effectively advancing drug discovery projects. So this is an example of some of the work that we do with partners. We certainly have all the expertise needed to do cloning and expression and stable cell lines. We have both plasmid vectors as well as viral vectors, and have, like many people, worked on iPSC-derived cells, mostly in our case neurons. We do assay development, and we have all the tools needed to do hit-to-lead identification and mechanistic screening. Our validated platforms for profiling include hERG. We run an enormous amount of hERG work for partners around the world, and we also have a cardiac panel for profiling, for in vitro profiling against cardiac targets. And what I'll talk about mostly today is our full panel of sodium channel assays. So since this is a pain webinar, I just thought I'd put in a couple of slides regarding the unmet need. And I think it's really important when you think about pain as a target that the current pain therapy, almost regardless of the drug that you're talking about, is guided by the side effect and tolerability profile. So patients are often not treated to the full efficacy needed, but only as guided by, essentially the paradigms used to treat the drugs. And there's study after study showing that, there's a huge unmet need in the pain community with patients not getting fully treated or poorly treated. I think we all know examples of friends and relatives that have had, pain states where it's been close to intolerable. It recently happened to my father. My father had a small fall. He's an elderly gentleman, had a small fall, and it took months of going to doctors to get the pain treatments right. So that's just a small personal example of what is a larger problem. The market is viewed at this point to be at least $10 billion, and there's an estimated 250 million patients that are undertreated. So that's the unmet need. So what would you do differently? So the key attributes, if you think about what the key attributes of the next generation drug needs to be, and you focus on neuropathic pain, really what you need to do is have an opportunity to develop a therapeutic that would be superior to the current, gold standard, those being Lyrica and Cymbalta. And while those are very good drugs, they're certainly only effective in about a third of the patients that need them. And building in, greater efficacy and improved safety profiling is what I think is very doable. So what's the way in? At the heart of every drug discovery project, you have to think about translational pharmacology. I could probably speak to an hour about the ability of animal models to be predictive, because in all therapeutic areas, there's always a level of uncertainty when you go from the preclinical space to the clinical space. And when I think about who's the person that I've learned the most from over the years, it's Sidney Brenner. And he had thought about this long before most other people. And part of what he says that has always resonated with me personally is how progress in science depends on new techniques, new discoveries. And those techniques and discoveries are usually the basis for the ideas that follow. And at Essen, we think exactly that way. We think about how hard, how we have to work very hard to make sure we're asking the right question with the right model so that we can generate decision-making data. And what I'll do in the next set of slides is give you an example of how we do that. So it's, I think, almost self-evident that there's a strong rationale for using ion channels as drug targets for treating pains. You could go through a much longer list than this, but for the sake of this seminar, I just highlighted the channels in the nociceptors that we think are the highest probability of success. And what I'll focus in on for this seminar is specifically the sodium channels. But if you look at CAB 2.2, N-type channels, and TRPA1, you could make a very good story for that, and it's something that we're working on as well, but we'll talk about today. So within the sodium channel world, the NAV 1.7 and 1.8 are good targets because they are intimately involved in the reception and transmission of the signals that generate through the nociceptors. And if you look at the sodium channel family in general, there's 9 members of the family that that have to be dealt with. So part of the problem with getting a drug that selectively hits that target is building in the selectivity against the other forms of the channel. The ones that people worry about most, of course, are NAV 1.5, which is the channel in the heart, so you need high selectivity of one. If 1.7 will be your target, we'll talk through that. You need high selectivity over heart for safety reasons, and for tolerability reasons, you need relatively high selectivity over the other channels. And if you think about the logic of from a not just from a morphology point of view and a physiology point of view, but from a pharmacological point of view, it's well established that relatively non-selective sodium channel blockers are very good in blocking pain cessation. The obvious ones being the local anesthetic. So you put a local anesthetic, in the right place, and you dampen pain. That's well established. So the trick would be to basically get a compound that works as effectively as a local anesthetic, but having it be an orally deliverable drug. There's a list of anticonvulsants, carbamazepine being the one that I'll exemplify here, that's been approved for trigeminal neuralgia. While the alpha-2 delta compounds aren't sodium channel blockers, I put them in there because they are anticonvulsants, and I'll talk about them later in the talk. What limits the utility of the present set of sodium channel compounds in pain is their side effect profile, just like all the drugs that are used to treat pain. And the ones, being, of course, the worst being that you have to build safety over cardiac, but also you have to look at tolerability in terms of blurred vision, drowsiness, ataxia, and nausea and emesis. So is it possible to go forward with a project where you get equal efficacy but less side effects? And the basis for that really comes from several venues. And the rationale for using 1.7, I think the rationale is best for using 1.7 as a drug target. And I'll just go through that quickly. 1.7 is, of course, located in the nociceptors. It's upregulated in various pain states. But more importantly is the human pharmacology. The human pharmacology of genetic mutations in 1.7 that really set the enthusiasm and excitement for the target. So there's gain-of-function mutations that are well known and involved in some but not all primary erythromyalgia. That's a condition where people find that they're in continuous pain. It's a terrible condition, but it sort of links an upregulation of the gain-of-function mutation of 1.7 to the perception of pain. The opposite is also true, and that's what makes it most compelling. So loss-of-function mutations, 1.7, are involved in a congenital inability to experience pain. One of the more famous individuals that had this was essentially a performer named Edward Gibson. He would go around and people would put sterile pins inside of him, and he would feel no pain. And when you look at how many people like this there are in the world, the estimate is there's about 750 of them. There's knockout data that suggests that in the animal models, the animal models data with knockouts of 1.7 support the human data. So that, in a nutshell, is the reason for being excited about 1.7. So what characteristics would you need for a drug that hit 1.7? Well, first and foremost, you would need selectivity, and I've gone through that. High selectivity over the heart under voltage control conditions that mimic the physiology of those conditions. So we think you'd need more than 30-fold. You'd also need selectivity over hERG, as you would with any project, and you'd need these compounds for increased tolerability to have limited or non-CNS penetrant properties or greater than 10-fold selectivity over the other forms of the channel in the heart. So but in addition to all of that, they have to be, these molecules would have to have other drug-like properties. They'd have to be Rule of 5 compliant, more or less. You could make trades there. They'd have to have relatively low protein binding, as all good drugs would have. And they have to have reasonable oral bioavailability and PK consistent with BID or QD dosing. So that's sort of a simple snapshot of what you need to make a drug in addition to the functional pharmacology you would need to have. And it's really a combination of all of that that would set the path for getting the next generation's pain drug using 1.7. So how do we do that here at Essen? So the first thing we do to build our foundation is we basically use all the tools in our toolbox to make sure that we start with reagents that are validated and stable. So all the cell lines that we make, and I'll show you examples of that, are made first by looking at the expression of the channel under manual patch condition, and to make sure that we get decent expression. And then we go through a rigorous cell line development exercise using our imaging tool, our imaging instrument, IncuCyte, which allows us to essentially develop the cell lines and under conditions that make sure that their growth and morphology are exactly the way we want them and unchanging. And that all then feeds into Ironworks, and most recently we're using Ironworks Barracuda to do a full 384 well analysis of the lines to get the functional pharmacology and cell line characteristics. So at the end of the day, what makes this work really well is that we're using Barracuda as the assay development tool as the cell selection tool for which all assays will be developed. And when we roll that into essentially, how it actually works day to day, we have Barracuda working here on a regular basis alongside of the original Ironworks systems that we had in place. Actually, the first Ironworks ever created in the world is still up and running, and we invite anyone to come visit us if they would like to see it. And using those two instruments, we can easily generate 100 data points per day per FTE, and that's more than enough to keep up with MedChem efforts. If you think about it from an IC50 curve generation, that's 100 IC50s a day. So that allows us to keep up with MedChem, but it also allows us to do focus library screening to generate the chemical substrate for the hit. And it positions what we do as far as sort of low, medium throughput screening all the way through efficacy and safety evaluation. So here's some data. So here is raw voltage traces of all of the NAV cell lines that we have. 1.1, 1.2, 1.3, which was expressed with beta 1, 1.4, which was also expressed with beta 1, 1.5, 1.6, 1.7, and 1.8 with beta 1. And in all cases, the stable cell lines that we have are stable for over 30 passages in continuous culture, and the whole cell currents are sufficient to build quantitative assays on high throughput electrophysiology platforms like Ironworks and Ironworks Barracuda. The next thing we do is go through the biophysical properties. This is an example of single hole recordings of each of our cell lines in a single hole situation, and so the biophysics are in keeping with the known properties of each subtype that we have. More importantly, I would say, is the pharmacology. More importantly to me, I guess, I'm a pharmacologist. Is all of that looks good, but what's the pharmacology? So this is just a short snapshot of tetrodotoxin sensitive. It's one of the earliest ways that the channels were characterized into TTX sensitive and TTX resistant members. And as you can see here, the blue traces are the currents prior to the addition of TTX, and the red traces are after, and all the TTX sensitive channels are in fact TTX sensitive, blocked completely by 1 micromolar TTX, and the two insensitive channels that we have, 1.5 and 1.8, are only slightly inhibited by up to 3 micromolar TTX. So at first glance, everything looks right. Then there's doing pharmacology with known reference compounds as the basis for looking at what we have and how we can improve it. And so here is a Here is an example of using a local anesthetic, in this case tetracaine, looking at it in a 25 pulse train protocol where pulses are, where each pulse is stepped from minus 80 to 0 at 20 hertz, at 10 hertz over 20 milliseconds. And what you're looking at is the activity of tetracaine at the first pulse here in the first slide on the left, with the IC50s generated from the first pulse versus tetracaine at the 25th pulse. And while there is, as you would expect, a use dependency built into, that's part of tetracaine, so depending on the subtype, you get a shift under these conditions of between 3 and 30-fold higher potency for the inactivated state. You can see that there is no subtype selectivity. And we have much more data behind this. If you would like to see that, we'd be happy to set up time to talk to you about. You could look at this a different way. You could look at this as a 2-pulse protocol, where you now look at the activity of the compounds at the resting state versus the inactivated state. And what you get is something relatively similar. So with the resting state, you get tetracaine inhibiting with an IC50 of 37 micromolar. And with the inactivated state, depending on the time, it goes down to, at 5 minutes, it's 2.6, and at 10 minutes, it's 1.9. Looking at everything, looking at all of our subtypes in the activated state, you see under these conditions, there's no subtype selectivity as you would predict. Compare that to a compound that's now very subtype selective. And everything that I'll show you in the next three slides are using the 2-pulse protocol. So looking at, so this was done, this slide was, the data on this slide was done in collaboration with AMRI, who is our partner in this. So the chemistry and the data mining was done at AMRI, and the electrophysiology was done here at Essen. So here's an example of a subtype selective compound where under in the inactivated state we're getting blocks depending on the time frame at 61 to 15 nanomolar, no activity on the resting state, and under inactivated conditions, high selectivity for 1,7 over the other forms. So the IC50 for 1,7 is, under these conditions, this is the 5-minute point, is about 61 nanomolar. Everything else is much greater, a big shift in potency. There's some minimal activity with this molecule on NAV 1.6. Compare this to another reference compound that we made. Here we're calling this compound 2. What you see in compound 2 is, once again, there's high potency on the inactivated state of 1.7, some activity on the resting state at higher concentrations. And now when you compare all of the members in our panel, you see that this compound is hitting 1.7, similar to 1.6. These are all human. These are all the human isoforms. I should have mentioned that. And so it's a selective blocker, but not as selective as the first compound. Minimum activity on the other subtypes, but it does hit 1.6. Compare that to compound number 3. Once again, potent blocker on the inactive state of 1.7, more block under resting conditions, but less selectivity across the isotypes. Most selective for 1.7, but clear activity at 1.6 and 1.3. We're not including 1.1 data because it's got an unusual effect on 1.1 that we're still evaluating, but minimal activity on 1.5 and 1.8. So when you roll that all together, and this is just an example, you put now tetracaine up to the other reference compounds that we have, you get the idea that with with the right chemistry, you can start dialing in different levels of, with the right chemistry and tools like the ones that we've developed, you can start building in subtype selectivity. And with the high throughput screening system that we have here on Ironworks Barracuda, we can turn the crank very, very quickly. So I'm not going to go into too much more detail because this is just an introduction. This is a plug for my friend and colleague Dave Rock. Dave Rock's going to be presenting a more complete picture of what we have at the Ion Channel Retreat, which is in about 4 weeks in Vancouver, Canada. So if any of you are going to be there, please look for Dave. He'll be telling you more about it there. And if you have questions, we'd be happy to answer that too. So okay, so that's the in vitro functional pharmacology that we can do very quickly. But that's only part of the story. The other part of the story is that these drugs actually have to work in more complete preparations. And what we think is the next preparation to use is something that we do in collaboration with a group in England called NeuroExpert. That's Tom Salt and Stuart Neal. And with them, what we think the next step is to basically take the NaV 1.7 blockers and look at their ability to block neuronal excitability using an isolated perfused model that they have. And then this would be the way of assuring that the drug-like properties that we think we're building in actually work in an intact in vivo preparation. What you're seeing here is just an example of data from Stuart and Tom showing the effects of TTX on the preparation. We would expect that the 1.7 blockers would have an effect on nerve excitability depending on the paradigm that we use, and that's something that's a work in progress. But it doesn't also end there. So recording the compound action potential from peripheral nerves is relatively straightforward and will demonstrate the ability of these compounds to decrease axonal excitability. But we would also test them in parallel in DRGs from both human and rats, and that would be a way of us ensuring that our recombinant cell lines are mimicking the functional pharmacology of the native tissue. What we can do from there is build on then translatable biomarkers by looking at in vivo assays for threshold tracking using microneurography. This is the sort of assay that can be used both preclinically and clinically as a way of measuring target engagement, which we think is absolutely essential for picking doses that are translatable from animal models to human, and something that we're clearly set up to do. So I'll just end by mentioning some of our thinking on gabapentin. So while this is not a sodium channel project, it's a project that's also built into thinking about ways of getting better drugs that hit the alpha-2 delta receptor. As those of you who may have studied this know, is that the alpha-2 delta subunit is one of the subunits of the calcium channels, and that gabapentin and pregabalin work through the alpha-2 subunit, but what happens after the compounds bind to it is hotly debated and poorly understood. This is just a slide I borrowed from my friend Charlie Taylor, who's been studying alpha-2 delta for a long time, and it goes through his idea that it's postulated that it modulates calcium influx under hyperexcited conditions. The data there is it's hard. There's some data, but it's hard to reduce. Again, reducing neurotransmitter influx is something that's been done, but hard to reproduce. And but what is easy to reproduce is that the binding site is central to the mechanism of action of the drug. That work was done by Jim Alford, who actually did the careful point mutations. And what this slide shows is that if you look at single point mutations in the alpha-2 delta 1 subunit, that's the R217A mutations, it doesn't change the expression of the channel, of the subunit of the channel, but what it does do is when you make that mutation in a knock-in mouse and you look at the ability of pregabalin to be an anticonvulsant, the black bars show the anticonvulsant activity of pregabalin in the wild-type mouse, and the gray bar shows its activity in the alpha-2 delta 1 knock-in mouse. And you can see it's largely abrogated. And that basically, that data set can be reproduced for anticonvulsant activities, it can be reproduced for nociceptive activities, and for anxiolytic activities. So it says that that binding site is very important. It doesn't say what the binding site does, but it links, it sets up a tool to then do the next more interesting experiment, and that is to build on anticonvulsant activity. You can set up the ability to do a phenotypic functional assay looking at what the alpha-2 delta compounds do in a leptiform slice model, and this is again something we're doing in collaboration with NeuroExpert. And the reason why this is of potential interest and another way in is that you can use the assay to determine the potency of the compound, and that potency should be reflective of its binding activity, so that's just a reality check. But more importantly, you can use it in a diagnostic way to look at the mode and the site of action within the neuronal circuitry. You could actually be able to drill in to start looking mechanistically at what's going on to give you another opportunity to find compounds that work better than the ones that have been previously used. And the key part of all this is that you would do the hippocampal slice preparation from wild-type mice and mice that have the knock-in mutation. And this is something that is available to us through a collaboration with Jim Offord and his colleagues at the University of Michigan. So I just put that out there as sort of a, as another example of the way we think we can work on new pain targets. So what's key to all of this is the fact that we do what we do well, and we look for other people who do what they do equally well. And when we combine together, we set up a strong partner network to be efficient and effective at prosecuting targets. And the network, it can be assembled to offer customized solutions to problems that are needed. I've given you 2 examples of that today. Here's our partner network. This is also on our website, so I won't dwell on it too long, but I will dwell on the last slide, which is to acknowledge the people that did the work. So all of us here at Essen believe in team-based science. We think team-based science is really, the way to be efficient and effective. And the members of Essen, that worked on this include members both in the U.S. and the UK. Here in the U.S., Dave Rock leads the effort. Owen McManus is the new hire, and we all work closely together. And the people who actually generate the data here are John Rock, Kevin Allard, Libby Opeka. Catherine Artimovich was central to doing all the molecular biology, and Dan Appledorn helped enormously in that regard too. Our colleagues in the UK work closely with the colleagues in the US. It's led by Del Trezise and his close colleague Tim Dale, and Srini Kamnu-Milli is also central to that work. AMRI chemistry was driven by Pete Guzzo, and NeuroExpert I mentioned before is Stuart Neal and Tom Salt, and Lori Eisen and Jim Offord at the University of Michigan were responsible for the alpha-2 delta side of the story. So I think at that point we're reaching the end of the seminar. I really thank everyone for their time and their interest, and there's some old friends that have logged in, and I'll say anonymously hi to everyone, and please get in touch. Thank you very much.
I'd like to thank you all for attending today's webinar. If you would like further information or would like to chat in general, please send an email to us at discovery@essenbio.com as indicated on the screen at present. Certainly personal emails can be sent to Vince and to any of us here at Essen as well. We would encourage you to check the Essen Bioscience webpage for further information about upcoming webinars. The next month webinar will be focused on our cell player angiogenesis assay using the IncuCyte cell player technology. So we encourage you to check that out as well. At this point, thank you very much for attending the webinar. We really appreciate your attendance today. Thank you very much.